Welcome to The Explainer, where today we're basically turning into garden investigators to solve the ultimate botanical mystery, plant pathology. Have you ever looked at a struggling plant in your garden and just thought, what on earth went wrong here? Seriously, plants can be incredibly dramatic when things go even a little sideways. Well, today we are going to systematically break down the clues, using the exact frameworks that plant experts rely on to diagnose and treat these green crime scenes.
Right, so picture this. You're standing at the crime scene of a failing plant. When you're suddenly faced with yellowing leaves or wilting stems, your absolute first instinct is probably to just guess the culprit, right? We all do it. But guessing without a real system often leads to unnecessary chemical sprays and, frankly, much worse problems down the line. Because without an accurate diagnosis, your treatment might not help at all. We really need a reliable method to figure out exactly why a seemingly healthy plant suddenly takes a turn for the worse.
Which brings us to Section 1, the botanical crime scene.
When we first roll up to this crime scene, we can't just start dusting for pathogen fingerprints right out of the gate. We've got to divide the world of plant problems into two very distinct categories. We always have to weigh our biotic suspects, and by that I mean the living organisms like fungi, bacteria, viruses, nematodes, and bugs, against our abiotic suspects. Abiotic stressors are the non-living environmental factors. Think drought, poor drainage, bad soil pH, a late frost, or even chemical injury from herbicides. In fact, experts actually advise that before you ever assume a disease is a contagious, living infection, you've got to rule out those abiotic causes first. They are incredibly common, usually much easier to correct, and hey, they don't require chemical pesticides to fix.
But hold on, before we even point fingers at abiotic stress, we have to ask a really foundational question. What is normal for this specific plant? People constantly submit samples of the white pine, totally convinced that this striking yellowing and dropping of needles in the fall is some severe garden-ending disease. But I promise you, it's perfectly normal. Every single fall, a healthy white pine naturally turns yellow and drops half of its needles, while the newer half stays completely green. So properly identifying the plant and knowing its natural quirks, that is step one of any successful diagnosis.
Moving right along to Section 2, decoding the disease triangle. So, what exactly gives our living, biotic suspects their motive and opportunity? Well, believe it or not, plant pathogens like fungi and bacteria are actually way less common than most gardeners assume. Infectious plant diseases just do not happen by chance. For disease to fully develop and rampage through your garden, it requires an absolute perfect storm. Plant pathologists call this framework the disease triangle, and it's the absolute core focus of today's explainer. It is a remarkably powerful diagnostic tool for literally anyone looking to keep their plants thriving.
Here is the crux of it. For the perfect botanical crime to happen, you need all three of these sides perfectly aligning at the exact same time. First up, you need a susceptible host. That's a plant that is genetically vulnerable or maybe just weakened by poor nutrition. Second, you obviously need the actual pathogen to be present and active. And third, you need a favorable environment. Think high humidity, weirdly warm temperatures, or soggy, poorly drained soil. Get this, even if a totally devastating fungus is hanging around in your garden, if the plant is highly resistant and the environment is nice and dry, the disease simply cannot take hold. All three elements must connect.
All right, Section 3, gathering your clues. With our disease triangle framework in our back pocket, let's look at how a smart detective maps the crime scene to narrow down that suspect list. The spatial arrangement of sick plants tells a huge part of the story here. If you see totally uniform damage, like say every single leaf on a plant, or an entire row of completely different species turning brown all at once, that screams abiotic stress. We're talking a sudden deep freeze, or maybe chemical drift. On the flip side, if the damage is random, scattered, or clustered in hotspots that slowly seem to spread, you are very likely tracking a living, biotic pathogen that's actively reproducing and jumping from host to host. Oh, and if the damage is in a perfectly straight line, yeah, that is almost always man-made. Think a rogue lawnmower or a trench that cut through the root systems.
Now, if our mapping does point to a living pathogen, we can quickly categorize them visually based on how they actually attack the plant's tissue. Fungi, which are definitely the most common horticultural pathogens out there, usually manifest as dry lesions with really clearly defined borders. Sometimes they even form these cool concentric rings. Bacteria, on the other hand, they are just messy. They cause these slimy, smelly, water-soaked spots with no real defined border. And viruses, they literally hijack the plant's cellular genetics. This results in weird colored streaks, twisted growth, and mosaic patterns. They are, for sure, the ultimate botanical hackers.
Which takes us right into Section 4, signs versus symptoms.
Let's dig a bit deeper into the physical evidence and decode the critical difference between signs and symptoms. This is a major stumbling block for a lot of everyday investigators, but it's absolutely vital. Signs are the actual, literal, physical presence of the intruder. If you can see spores, fungal mats, bacterial ooze, or insect frass, you are looking at a sign. Symptoms, however, are how the plant's body is reacting to being attacked or stressed. Things like wilting, yellowing leaves, or dropping fruit. Those are symptoms.
Check this out. This brilliantly illustrates the difference. On the left, we see bright orange rust pustules erupting right out of the plant tissue. That is a clear-cut sign you're looking directly at the reproductive structures of the rust fungus itself. Now contrast that with the image on the right. That black, dead tissue is called necrosis. You aren't seeing the actual pathogen there at all. You're just seeing the symptom of the plant's tissue dying off in response to stress or infection. Here's another perfect example for you. On the left, this cucumber leaf is covered in a white, fuzzy mat. That's powdery mildew. And because we can see the fungal hyphae and spores right there with the naked eye, it's a sign. But over on the right, we see these dry, tan spots bounded by the veins of the leaf. This is a localized symptom. The plant is desperately reacting to an invader, but we can't actually see the invader itself just by looking at the spots.
To be a top-tier detective, you really need to speak the language of symptoms. When you see rapid discoloration and death of foliage, we call that blight. When leaves turn yellow, which is usually from poor nutrition or root issues, that's chlorosis. A canker is a sunken, discolored dead area on a woody stem. And wilt? That's that general drooping caused by a loss of water pressure. It could mean drought, or it could mean a pathogen is literally clogging up the plant's internal plumbing. Let these specific symptoms guide your investigation naturally, rather than trying to force some predetermined diagnosis to fit what you want to see.
But sometimes, the suspect just leaves a very messy, totally undeniable clue. Look closely at this branch. We've got a canker, a sunken, dead area on the bark, which we know is a symptom. But look at what is coming out of it. That sticky amber liquid is bacterial ooze. Finding this is literally like finding the smoking gun. That ooze is a direct, undeniable sign of the pathogen. It absolutely proves that the internal transport system of the stem has been compromised by a bacterial infection. And worse, that ooze can easily be splashed by rain or carried by bugs to infect your neighboring plants.
Let's wrap this up with Section 5, Breaking the Triangle.
So, as garden investigators, we've gathered our clues, we've distinguished abiotic from biotic, and we've accurately mapped our signs and symptoms. Now, how do we use all this knowledge to solve the case and actually protect our plants? We bring it all right back to the disease triangle. The most empowering realization in all of plant pathology is that we don't always have to completely eradicate the pathogen to save our garden. Pathogens are just a natural part of the environment. Instead, we just need to strategically disrupt the other two sides of the triangle. If the host isn't susceptible or the environment isn't favorable, the disease just collapses.
And here's how you actually execute that strategy in the real world. First, pull the susceptible host completely out of the equation. You do this by intentionally choosing disease-resistant or tolerant plant varieties for your space. Next, you can go after the pathogen itself, but not just by blanket spraying chemicals. Rigorous sanitation is your best friend here. Disinfect your pruning tools between cuts and sweep up diseased debris so the pathogen has absolutely nowhere to overwinter. Finally, destroy that favorable environment. Something as surprisingly simple as spacing your plants out to improve airflow or watering right at the base of the plant instead of overhead can work absolute wonders. You instantly eliminate the high humidity and wet foliage that fungi and bacteria so desperately need to reproduce.
Now that you can read the spatial clues and truly understand the intricate dance of the disease triangle, the power is entirely in your hands. You aren't just guessing anymore, you have a legitimate scientific diagnostic system. But it really makes you wonder, if changing just one tiny local environmental factor can stop a pathogen dead in its tracks right in your backyard, how are shifting global climates rewriting the rules for plant diseases worldwide? It's a massive, fascinating puzzle. But until next time, as you step out into your garden and spot that very first yellowing leaf, ask yourself, which side of the triangle will you break today?